Hi guys, I'm back. All right, let's do this one here. Share screen. Okay, share. All right. So we talk about this. We are going to talk about this membranous organelle. So here, list of them. Here I highlight that nucleus and mitochondria has double membrane. Means that typically, um, like a cell has single membrane. But in the nucleus, it's very much like this one here, very much like this one here. So it has a double membrane. Okay, it's this one here, this one here, this one here, this one here. Okay, so it has double membrane. Mitochondria also has double membrane, it's outer membrane and inner membrane. We will talk about those. So nucleus, not all cells. Let's start by talking about nucleus. Not all cells has nucleus. Red blood cell doesn't have nucleus. Typically, it has one nucleus. Skeletal muscle cell has multiple nuclei. Okay, its plural is nuclei. Nucleus. The function of nucleus is to contain the genetic information. So it's a chromosome. Typically, somatic cells has 23 pairs of chromosome. And so each nucleus has two membrane, double membrane, and this one is called the nuclear envelope. So that's right here, nuclear envelope. And there is a nuclear pore. So this is nuclear pore. Things can go in and out. The thing go in and out is the RNA. So this is nucleus, example of nucleus. Here you can see a lot of chromosome. The center portion, it is the nucleus. Nuclear pore, things can move in and out. And you can see that it actually attached to the endoplasmic reticulum. So the outer portion of the membrane extend to the endoplasmic reticulum. And attached to that is the ribosome. So cells, the chromosome number in the somatic cells, the chromosome number is 23 pairs. So like this one here, so that is one pair, that is another pair. So one's from your father, one is from your mother, and that's one from your mother, father, one from your mother. And so that's somatic cells. Germ cells is one that only has one pair, it's not a pair, but only one of these chromosomes. So it's randomly split from this pair, and so this one is called haploid. It only has 23 chromosomes, not 23 pair, but 23 chromosomes. And because it's randomly split from one of these pair, one part of this each pair. So this will produce each of these germ cells to be unique from each other. These somatic cells are all duplicated from the fertilized egg. So they are all the same. They have the same sets of chromosomes. So we have this germ cell with only haploid will mate with another germ cell, which is also only 23 chromosome, and to produce a fertilized egg. That fertilized egg can be then through this mitosis, and that will be duplicated to produce an entire body. And then with that entire body, it will also then produce its own germ cell, a lot of like germ cell. One of it will mate with another person's germ cell, and then to produce the fertilized egg. Okay, so this one is an example that one of the germ cell from one sperm with the egg mating, and then it will produce this fertilized egg. Then this fertilized egg has diploid 23 pairs of the chromosome. Then that will, through the mitosis, that will produce the entire body. So all our cells in our body have the same sets of chromosomes. So this one is example that we have 23 pairs of the chromosome. Each one of these pairs we have 22 autosomes, means that these pairs are not related to sex. But we have one pair coming from one from father, one from mother, and the combine of this XY, XX would define the person's sex. So this is a sex. This pair is a sex chromosome. So overall, we have in the somatic cells, we have 23 pairs of chromosome. So that's located in the nucleus. So we talked about the chromosome, and earlier we talked about this DNA. So what is the relationship between the DNA and the chromosome? So basically, that DNA, as we mentioned earlier, that DNA is a polymer. So each one is a polymer of the nucleotide. So you can see that it linked together. So the entire length is the DNA. This is the DNA. A segment of that is a gene. So for example, that from here to here is one gene, for example. And that gene is used to code RNA and to use to code protein. So that is one gene, and then the entire DNA may contain hundreds of thousands of different genes. One may be used to define your hair color, your skin color, your eyes color. So that's a DNA, and we have a double-strand DNA. Then what is a chromosome? Chromosome basically is that this DNA will have to be folded around a protein. This protein is called histone. So that's the histone protein wrapped around the histone. So, and then the entire thing is called chromosome. So chromosome is not just DNA, but DNA plus protein. The major protein is histone. So with that, we can have a gene expression from the DNA transcribed to the RNA and then translate to the protein. RNA will coming from the nucleus flowing out, and then RNA will go into where? Ribosome, yes. And where over there, we can have the translation from RNA into protein. So here is the picture showing you that we have the RNA polymers to transcribe DNA into the RNA. There are several types of RNA, but messenger RNA is the one to be used to produce protein through what? Through this ribosome. Then it's produced the protein. And what's the, so just a little bit like a overview about what is gene, what is the chromosome, what is the DNA, what's their relationship. So as I mentioned before that DNA has very long, is a long like polymer, like this one here. And the one segment of it is a gene. So this segment is a gene, is one gene. And this gene is used to code a specific protein, a particular protein. So that is one gene. The location of each gene is called locus. So it's right here. Location of each gene is called locus. And each person has one pair of the chromosome. So this gene, each person has two basically duplicated, has one pair of gene, one from your father, one from your mother. The color of the eye can be defined by mother and father. So depending on which one is dominant, then it will be expressed. So this is called an allele. Allele is a variant of the same gene. So this is the same gene, but two variants. And if in the whole population, we may have multiple variants of the same gene. Like hair color, we have so many different kinds of hair color. It's all located in the same locus. It's the same gene, but some variation will make the person express different feature. The size of a gene can be varied, like I mentioned before here, that this is one segment, right? So say this one has only like 10 base pair of the DNA, and that is one gene. But some gene can be very long, so it depends on it really varies a lot. So the size of the gene can be varied out, ranging from 76. You don't need to remember the number. You just need to know that the size of a gene can be varied a lot. So we have a human has a lot of DNA, a large number of the DNA. One thing is that not all this DNA is used to express for protein. Only about 2% of them is used to express protein. So there are a lot of DNA is used to produce the other RNA that's for the regulation purpose, but not specific to be used to produce protein. So you need to know the term, what's the gene, what is the locus, what is allele. Okay, so that's the topic here. And then the outside of this outer membrane of the nucleus is the endoplasmic reticulum. And we have the smooth ER and the rough ER. On the rough ER, we have this ribosome. Ribosome is that the organelle to produce protein. So ribosome, as I mentioned before, that ribosome can have a free and a fixed, right? It's a non-membranous organelle. If it's fixed, it's located in the ER, it's right here. And it produced protein will be packed in the ER, snitched off in the vesicles, and be transported. So this is the function of, so rough ER has these ribosomes to produce the protein. Smooth ER contains a lot of this material to produce. So smooth ER, basically this one content, not this one, but here you have this. These are all like a lipid bilayer, right? This is good material to provide this package material for vesicles. So they produce lipid bilayer to produce the vesicles for the protein transportation. And then these vesicles will then move to the Golgi apparatus. The function of Golgi apparatus is to fold this protein, because once the first is produced, it's a linear structure, but it has to be folded in order to produce this protein. And then these vesicles will pinch off from the Golgi apparatus, and then it will be released to the extracellular space. One example is that this one is, say, insulin, right? So insulin is a protein, is an important messenger to opens up the door for the glucose in other cells, right? So then it will be first has to be transcribed from the DNA, translated to the protein, packed in vesicles, and waiting here. When the glucose level is high, then it will be released. So just a little like these vesicles, as we mentioned before, basically is the same material as the plasma membrane, as the cell membrane. It has phospholipids around it. And basically the same membrane structure of the cell membrane, of the endoplasmic reticulum, and the plasma membrane. They are basically the same. And we have this endocytosis, right? We mentioned before endocytosis. It has a receptor, binds on it, engulf it, bring it in, or the exocytosis. So it moves out there and secreted. So this one is the combination of the, then let's talk about we talk about the Golgi apparatus, then let's talk about the lysosome. Here is an example that showing you that we show this figure just earlier, right? So the material from the ER move to the Golgi apparatus, and then this material will be either released into the extracellular space, or they can be packed in the vesicles and merge with the lysosome, and they provide the enzyme, help this lysosome to digest this material. So let's talk about this lysosome. Lysosome is an important digestive organelle in the cell. Its function is to break apart, break down, conduct this catalysis, catalytic process to break apart large molecule into smaller molecule, break apart the protein through the hydrolysis. That's the term I was looking for. Catabolic process. Protein is hydrolyzed into the amino acid, from the polymer into the monomer, glycogen to glucose, and lipid to the fatty acids and glycerol. So basically, it's from the polymer into the monomer. Just another picture of Golgi apparatus, right? So here we show that substance can packed in vesicles, move to the Golgi, and then this basically is protein, move to Golgi. You can pack it and fold it, modified it, release it into the extracellular space, or they can be merged with the lysosome to provide the enzyme to digest these large molecule. So we talk about these Golgi apparatus that basically release these vesicles, and that can be either secreted into the extracellular space or merge with the lysosome. So it can be the secretory vesicles or the lysosome vesicles, like this one here. So that's Golgi, and another one is Paroxysm. Paroxysm coming from the smooth ER, and the function is to break apart long chain fatty acids. So we talk about that fatty acid has this structure, right? And this chain can be very long, so it could be a long chain. Sometimes long chain can be more difficult for our digestive process to break apart. So here, paroxysm can conduct that function to break it apart. Paroxysm also can be produced by fission, like one paroxysm can be split into two paroxysms. So it's called fission. So the production of paroxysm can come from the smooth ER or coming from the fission process of one single paroxysm. And the next one is the mitochondria. So this is a typical EM. If you look at the microstructure imaging, you can see mitochondria very typically like this one here. And mitochondria has, the major function of the mitochondria is to produce ATP. So when you look at the mitochondria, first thing you need to think about is the ATP. So the major function is to produce ATP. And why do we need ATP? Where do we see the needs of the ATP? One region that cells need ATP all the time. Even if you don't do anything, you burn ATP. The reason is that we have this sodium, potassium ATPase. So remember that in a cell, we have this pump. What? Pump what? Yeah, 3 sodium. Pump what? Into potassium, okay? So this pump requires ATP. So even though you don't do anything, you need ATP. If this doesn't have the ATP, the cell would die, and that's not a good thing, okay? That means ischemia. That means that's a consequence of the stroke, that you don't provide enough blood flow into the brain, for example. Then the brain doesn't have enough oxygen. Oxygen is required for the oxidative phosphorylation to produce ATP. And also that if the blood doesn't provide the brain, there's not enough glucose. The brain neuronal cell require glucose, and then it cannot produce ATP. Then the cell will die, okay? And that is a consequence. Very quick, a cell cannot live for a long time without continuous supply of oxygen. So that's why we need ATP. Even if you don't think anything, if you just playing game or something like all day long, to keep it alive, you need energy just by keeping it alive. So do something, use your brain. It's there. It's you already supply it. Why not using it, right? Okay. All right. So this is mitochondria has a double membrane, two membrane, outer membrane. So let me go through that terminology first. First, we have the term called outer membrane. This is very not important. Outer membrane, not important at all. What's more important in inside? So what's more important is inner membrane. Okay, here we have this one is called inner membrane. Almost everything happens here, inner membrane. The space between is called intermembrane space. So here, the space here is called an intermembrane space, between membrane. And intermembrane space has a lot of H+. We will learn about it. And inside of this inner membrane, inside of this inner membrane is called inner cavity. So here is the inner cavity. This one contains matrix. Do we have term here? Yes. Inner cavity, sorry. Inner cavity is right here, inner cavity. So this is inner cavity. This one is also contains a matrix. Matrix contains a group of proteins and enzymes, basically. Here contains a lot of enzyme protein. This enzyme is for the oxidative phosphorylation. Okay, so this is the region where we need the oxygen. So that's that. The key here, the key player here is the inner membrane, the matrix, and the intermembrane space. Okay, so that's the key. And the function overall is to produce ATP. So let's look at what it does. Do I record? I hope I did. Yeah, I'm recording. So the function is to produce ATP, and the very basic process to produce ATP is called ADP phosphorylation. Means that ADP has D is di, means that it has two phosphate. So phosphorylation means add another phosphate. So here is ADP, add another phosphate to make it ATP, become three phosphate. Here is two phosphate. So that's the product process, very simple one. The whole work is to add a phosphate to ADP to become ATP. Through this process, we need oxygen, and that's why we call it oxidative phosphorylation. So let's see how mitochondria does it, okay, shall we? Phosphorylation adding a phosphate, this is common mechanism to regulate protein function. So sometimes we have protein, we can add phosphate to it, phosphorylation process. But this is also a process here to produce ATP. This is not protein, this is a nucleotide. And add phosphate to it, we can produce ATP. So how does mitochondria does it? Overall, what it does, the end process. Let's look at from the end first, the final. So we get the idea that that's how it's produced. So this is very last process to produce the ATP is right here. Right here. So this is what we have talking about. ADP phosphate to become ATP. So this is a phosphorylation. How does it do it? Here is the location of the inner membrane. As I told you before, inner membrane is a key place that these proteins are called electron transport chain right here. This is an electron transport chain. They are located in the inner membrane. And so they are located in the inner membrane, and so what it does is that overall that the whole process is to generate high concentration of hydrogen in the intermembrane space right here, intermembrane space. And that hydrogen will diffuse through this channel. So this is very much like a windmill or something that blows through it. When it blows through it, that energy can convert ADP to ATP. So that's a process. Hydrogen goes through it, make it the move. It's like windmill. When wind blow it, the mill work to produce ATP. And the question now is that how can we have so many hydrogen here? The reason that we have so many hydrogen here is we have two important players. That is NADH and FADH2. NADH will donate its hydrogen to here to become NAD+. FADH2 will also donate his hydrogen here to become FADH, FAD, not H, FAD. And so this one is an important contributor to contribute their hydrogen, into this intermembrane space. And that accumulated hydrogen goes through this channel to produce ATP. So that is the end process. With this one though, we will need oxygen. The reason is that when they contribute this hydrogen, they also release an electron. So the electron will pass through this electron transport chain and be reduced by the oxygen from our breathing. So without it, we cannot. And the production of it is the H2O. So this one will produce H2O. So we need oxygen to receive this electron. And the major player here is the NADH and the FADH2. Okay, so it's located. So this one is called, the process is called oxidative phosphorylation. The reason it's called oxidative is because we need oxygen. And this process, the contributor is NADH and FADH2. So the question is which two molecules provide their hydrogen in the process of oxidative phosphorylation? The answer will be NADH and FADH2. And the question is where is hydrogen is stored? Where the hydrogen is stored in the intermembrane space. And where is the electron transport chain? Where are these proteins? They are located in the inner membrane. So those are the questions you will see in the quiz or exams. So the electron transport chain works because we have this NADH and FADH2. They contribute their hydrogen, and the hydrogen concentration provide that concentration gradient to push through this channel to produce the ATP. And through that, we need oxygen to receive this electron. And this process generates a proton gradient. This proton gradient means hydrogen gradient because H plus is also a proton. Go through this one here to produce ATP. So that's that. Easy, right? Very easy. Okay. Problem solved. Now let's answer a very final question. Because we already know that how mitochondria produce ATP, very simple, just in the inner membrane, hydrogen accumulated, go through it, produce ATP. This one requires oxygen, that's easy. But one thing we have not answered is that where is NADH and FADH2 come from? Where do they come from? We need those to contribute hydrogen, but where do they come from? They come from this further process in the matrix. This process is called citric acid cycle. So citric acid cycle will produce NADH and FADH2. It's not showing here, but it will produce these two. And so let's look at this citric acid cycle. So this citric acid cycle is a process in matrix. Sorry. Okay. This is process in matrix. Okay. And here basically is the process to produce NADH and FADH2. And we will use that in the electron transport chain to produce the ATP. And so that's how they are produced. Their source though, coming from the glucose. So if we have the glucose here, glucose will first convert through the glycolysis to produce these pyruvate. And then pyruvate will produce the acetyl-CoA, enter into this process through this cycle, and then it will produce lots of the NADH and FADH2. Through this cycle, though, we will also produce CO2. That's why we need to breathe out CO2. CO2 is produced in the citric acid cycle. O2 is needed in the electron transport chain. Right here, O2 is needed here in the electron transport chain. So even though we do need to breathe in O2 and release CO2, the the region that we use O2 is different from the region we produce CO2. The region we need O2 is in the inner membrane, the electron transport chain process. The region we produce CO2 is in the matrix through this citric acid cycle. So that's that. In the matrix, we conduct this Krebs cycle, Krebs citric acid cycle, and that the end product is the NADH and FADH2. Then we will use it right here in the inner membrane to use them to contribute hydrogen, build up this hydrogen gradient or proton gradient, proton concentration to push through this one. And then that will produce ATP. The reason we need to breathe, the reason we need oxygen is in the inner membrane. The reason we need to breathe out CO2 is in the citric acid cycle. The reason we need to eat is because citric acid cycle, we need the source coming from the glucose. When they enter into the matrix, it's converted into the acyl-CoA, and that goes through this process. The end product, the needed product, is NADH and FADH2. So that's that. Citric acid cycle is also called TCA cycle, also called Krebs cycle. So you can hear these terms kind of interchangeably in different area. The location is key though. You need to remember that they are located in the matrix of the mitochondria. From each glucose, we produce about 10 NADH, 2 FADH2. Generally speaking, each NADH can produce 3 ATP. Each FADH2 can produce 2 ATP. So overall, we may produce about 36 ATP from each glucose. So that is a very full use of each glucose. So oxygen is needed. That's why we call it oxidative phosphorylation. And where does oxygen needed? It's in the electron transport chain. It's used to receive that electron. And the foodstuff, right? Like glucose. We use glucose as the example. It's more direct, that glucose to be converted into the acetyl-CoA is more direct process. Fatty acids can do that too. Amino acids can also produce, can also enter into the citric acid cycle, but it's not as direct as the glucose. So a major one is still glucose. The other can, but it's not as direct. So glucose go into the mitochondria, produce 36 ATP. If it doesn't enter into the mitochondria, if there's no oxygen, then it's the glucose glucose will only produce 2 ATP. So if there's no oxygen, this process is called anabolism. Anaerobic process with no oxygen. If there's oxygen, it's aerobic process, right? Anaerobic process. With that, we only produce 2 ATP. So it's not efficient. It's very not efficient. So overall, that inner membrane has a lot of this folding. It's called cristae, right? A lot of this folding, and that folding allow a lot of this space to conduct the oxidative phosphorylation. And hydrogen is built, pushed through to produce the ATP. Hydrogen is accumulated in the intermembrane. So that's the major function of the mitochondria, to produce the ATP. One thing we need to know that mitochondria contains DNA. Mitochondria has its own mitochondria DNA. So in our cells, technically speaking, two organelles contain DNA. One is our nucleus, the other one is the mitochondria. These two contains DNA. Mitochondria DNA coming from mother only, because mother eggs has the, everything there. Father sperms need to only focus on moving, so a lot of these extra organelles are removed. So we got our mitochondria directly from the egg, and that's where we got the mitochondria DNA from mother only. Mitochondria can be produced by fission, just like what we mentioned about the paroxysm. So it can be break apart, 1 become 2, 2 become 4. Each one can also grow. So that's it. Is that it? Yeah, good. Thank you so much, guys. All right, I will prepare for the next one and upload it very soon. I think this one will end the first week. So getting there.